Hello everyone, welcome again to my YouTube channel. So today we are gonna do is carbohydrates. Okay, so in this part of video we will discuss about the general structures, all the general structures of carbohydrates, and every important point which can be asked in uh, any kind of competitive exam or any kind of university exam. So we will try to cover all the important points, whatever could be possible. Okay, so without wasting our time, I think we should start our this part of video. So let's move towards the structures of carbohydrates and also the classification of all carbohydrates. Okay, so let's start. Firstly, the point arises is what are carbohydrates? Carbohydrates are polyhydroxy aldehydes or polyhydroxy ketones, or the or compounds which produces them when they get hydrolyzed, or on hydrolysis, which produces them on hydrolysis So let us try to understand what is polyhydroxy aldehyde or ketones. Okay, so polyhydroxy aldehyde, let's try to understand this. Polyhydroxy means more than one. Okay, polyhydroxy means more than one. Hydroxy is the OH group and aldehyde group is this one. Okay, when a compound is having more than one hydroxy groups and one aldehyde group, okay, or when a compound is having more than one hydroxy group and one ketone group this is the ketonic group. Okay, and this is the aldehyde group. Okay, so for example, let's try to draw a ketone. Okay, or a polyhydroxy ketone. So you can see CH2OH, C double bond O, CHOH, CHOH, and finally you can see here is a ketone group and more than one hydroxy groups. Okay, so on the basis of the number of carbons and on the basis of the position of these hydroxy groups, these ketones are further classified their specific name. Okay, so in the same way, there will be an aldehyde. The aldehyde group will be present on the terminal end of the that compound. Okay, so it will be something like this. So see, here is the aldehyde group and here are more than one hydroxy group. So these are the carbohydrates. Okay, so now we saw the carbohydrates. Okay, so now we will look forward to the about the classification of carbohydrates. The actually the carbohydrates are classified mainly in three categories on the basis of number of sugar units attached to that carbohydrate. Okay, so here is the classification. So firstly, the in three major groups the carbohydrates are classified, and on the basis of number of sugar units, on the basis of number of sugar units. They are classified into three major categories. The first one is monosaccharide, the second one is oligosaccharides, and the third and last one is polysaccharides. Polysaccharides. Okay, so the monosaccharides, let's talk about the monosaccharides. Monosaccharides are also known as simple sugars. Simple sugar. Simple sugar. Why are they called as simple sugar? Because they composed of only one single sugar unit. Okay, oligosaccharides are composed of 2 to 10 monosaccharide units. Monosaccharide units. These are oligosaccharides, and polysaccharides composed of more than 10 monosaccharide units. Monosaccharide units. Okay, so these were the monosaccharides, oligosaccharides, and polysaccharides. Okay, so the next characteristic of monosaccharide is they can't be further hydrolyzed. They can't be further hydrolyzed, whereas these all can be further can be hydrolyzed. Hydrolyzed. Okay, so uh, this can be further hydrolyzed. So as we talk here that oligosaccharides are composed of 2 to 10 sugar units. So on the basis of these number of monosaccharide units, they are further called as, if they are having 2 monosaccharide units, then they are called as disaccharides. Then they are called as disaccharides. Okay, if they are having 3, then they are called as trisaccharides. And in the same way, tetrasaccharides, pentasaccharides, etc., etc. Okay, so the most important group of oligosaccharides is disaccharides, which are like we should focus on them. Okay, so I'll give it a 3 star. Okay, so the most important sugars are the disaccharides, which we will have to do or learn by heart. Okay, so and polysaccharides are also classified into two main categories, into two types. Polysaccharides are of two types. The first one is Homo Polysaccharides, Homo Polysaccharides, and the second one is Hetero Polysaccharides. Okay, so now we will look separately. Firstly, we will do monosaccharides. Then in oligosaccharides, we will only discuss disaccharides, because disaccharides covers a very important part. Okay, so we will focus on disaccharides, and disaccharide is the most important part in carbohydrates. Then disaccharides, and then further we will move towards polysaccharides, in which we will see separately homopolysaccharides and heteropolysaccharides. Okay, so I think let's move towards now to monosaccharides. The first one is monosaccharides. Okay, so monosaccharides, when we talk about monosaccharides, on the basis of the group, on the basis of group or functional group or functional group which is attached to them, functional group, they can be classified into two main categories. Okay, the first one is aldoses, the first one is aldoses, and the second one is ketoses. Okay, second one is ketoses. They are called as aldoses when they are attached to an aldehyde group. This is an aldehyde group. And when they are attached to a ketone, okay, group, so they are termed as, or when they are having a ketone group or an aldehyde group, so on the basis of this they are termed as aldoses or ketoses. So let's cover them in a box. Okay, so now they are also classified on the basis of number of carbons, on the basis of number of carbons, on the basis of number of carbon atoms. They can be also named as, like, if they are having 3 carbons, 4 carbons, 5 carbons, they are having 3 carbon, 4 carbon, 5 carbon, 6 carbon, or 7 carbon. Okay, so now a question may be arise in your mind that why did I just wrote firstly 3 carbon sugar, because and why didn't I write 2 carbon or a single carbon? If I write a single carbon, so it's not an carbohydrate, it's an aldehyde or a ketone. Okay, so 1 carbon will be a aldehyde, or either an aldehyde, either a ketone. Okay, and now you'll say that okay, 1 carbon will be an aldehyde and 1 carbon will be maybe a ketone. Okay, but what's the issue with 2 carbons? Okay, See, I in the starting I told you the definition of carbohydrate. The carbohydrates are polyhydroxyaldehydes. They should be an aldehyde. See an example. They should be an aldehyde. Okay, and let's say I draw aldehyde and with a two carbon chain. Okay, and a ketone. Do you think a ketone is possible with one carbon or two carbon? A ketone is not possible with one carbon or two carbon. Ketone is only possible when it's having more than two carbons. Then it's a ketone group. Okay, but when it's having like less than two or two carbon, it's an aldehyde group. See, if I am trying to draw a aldehyde group, so a ketone group, the oxygen is either present here or on this carbon. Okay, so if it's attached here or here, one hydrogen will be here, one hydrogen will be here, so it's an aldehyde group, not not a ketone group. Okay, and that's not the point to discuss. The point to discuss is why I didn't say call the two carbons as a sugar. So if I'm drawing a two carbon chain with an aldehyde group, or the ketone are not possible with two carbons, so let that point, let's talk for two carbon aldehyde. So there will be only one hydroxy. Okay, so it's not, there is only one hydroxy, but the definition call that those aldehydes which are having more than one hydroxy group are known as carbohydrates. But here is only with two carbons, only one hydroxy group is possible. Okay, with two carbon carbohydrate, only one hydroxy group is possible. That's why they are not called as carbohydrates. So the most simplest carbohydrate ever possible is of three carbons. Okay, so the three carbon carbohydrate, we call them as triose. Okay, the four carbon is called as tetrose, and the five carbon carbohydrate is or monosaccharide is called as pentose. The six carbon monosaccharide is called as hexose, and then further heptoses. Okay, heptose. So okay, let's take an example. So for example, we would like to name glucose. Glucose is a six carbon sugar, okay, which is having a aldehyde group on the terminal end of that carbon chain. Okay, so the glucose is a six carbon sugar. The glucose is a six carbon sugar, is a six carbon sugar, and having an aldehyde group. So it is also known as aldohexose. Okay, in the same way, if I take an example of fructose. Okay, fructose is also a six carbon sugar, but it's having a ketone group with it. Okay, so it's known as ketohexose. Understood? So the next point arises is the general formula of carbohydrates. The general formula of monosaccharides. The general formula of monosaccharides. The general formula of monosaccharides is CnH2ON. Okay, CnH2ON. What you can see here, see, there are equal number of carbons and equal number of water molecules, okay, in monosaccharides. In monosaccharides, equal number of carbon and equal number of water molecules. So if you want to see how many, like if you want to draw the number of structure of any carbon, any monosaccharide, so you just need to know how many number of carbon it contains. For example, let's take another example. uh, let's take an example of glucose itself. See, glucose, we just know that glucose contains, we just know that glucose contains 6 carbons, okay, so 6 carbon it contains. So and we need the general formula of glucose. So uh, 6 carbon, so the value of N is equal to 6. So the general formula of glucose will be C6H2O 6, and if we try to calculate it, it would be C6H12 and O6. This is the general formula for glucose, okay. So it was about monosaccharides. Now we will look forward about those trioses, tetroses, the examples of which sugars are included in trioses, tetroses, pentoses, and hexoses as well, okay. So let's see the first one, which is trioses. Trioses. The trioses are 3 carbon sugars, okay. The trioses are 3 carbon sugars, and their example will be DHA, means dihydroxyacetone, PGAL, and DHAP. Okay, don't be confused, I'm telling you the full form, okay. I'm trying to write here the full form also. DHA is dihydroxyacetone, okay. PGAL is phosphoglyceraldehyde, and DHAP is dihydroxyacetone phosphate, okay. See, these two sugars are like very important these sugars play a very important role in glycolysis in the which is a step of carbohydrate metabolism, okay. So these sugars are very important, so you need to remember the 3 carbon sugars which are formed during carbohydrate metabolism, that are PGAL and DHAP, okay. PGAL means phosphoglyceraldehyde and DHAP means dihydroxyacetone phosphate, okay. See, in this part the question that would be asked in any kind of competitive exam is just a simple question that they are gonna give you the name of sugar and they're gonna ask you which one is polysaccharide, which one is disaccharide, which one is monosaccharide, okay. So the only this kind of simple questions can be asked, okay. And other question about their structure also can be asked. We will see the important sugars which are very important with their structures, okay. So this was about trioses. Now move towards tetroses. The second one is tetroses. Tetroses. Okay, the tetroses are 4 carbon sugars. Okay, tetroses are 4 carbon sugars, and the example of tetroses is erythrose 4-phosphate, the only 4 carbon sugar which is used in plants. Okay, in Kelvin cycle it is used in plants. Okay, so the example of 4 carbon sugar is erythrose 4 phosphate. Okay, so erythrose 4 phosphate, and please learn by heart all the examples which I am giving them star or 3 star or any kind of stars if I am giving them. Okay, so they play a very important role and you need to learn them by heart. Okay, so it was about tetroses. So let's move towards the next one is pentose. Pentose sugars. Okay, the pentose sugars are very important for us. Okay, and plays a very important role. Okay, so pentoses are 5 carbon sugars. Pentoses are five carbon sugars, and the examples of these sugars will be ribose, deoxyribose, xylose, xylulose, and arabinose, and also arabinose sugar. Okay, these are the examples of pentoses, but the most important the example I'd like to say is only ribose, the 5 star point. Okay, and the 5 star is deoxyribose. Their structure is also important, but we have discussed their structure in our previous video, which is nucleotide part 1. Okay, and we explained their structure and their importance, important roles which they play. Okay, okay, I'll tell you, they play a very important role in the formation of genetic material, DNA or RNA. Okay, so they are included in formation of them. So these are the very important sugars. Okay, so these are pentoses, ribose and deoxyribose sugars. Now we move forward towards the 6 carbon sugars. 6 carb, oh sorry, let's move towards 6 carbon sugars, which are hexoses. Okay, hexoses are, see, I'm 6 carbon sugars, 6 carbon sugars. Okay, so the example of 6 carbon sugars is fructose, fructose, glucose, galactose, mannose. See, it's quite complicated to learn. Okay, so you need to learn them by some trick or some kind of and any kind of trick you can make for them. For example, I just did and firstly, please learn hexoses by your heart. Hexoses play a very important part in competitive exam, and also these are very important sugars, and especially the fructose and glucose. Okay, because they play a very important role in carbohydrate metabolism, and the in the carbohydrate metabolism cycles, they act as the substrate. Okay, so and provides the energy instant energy source, okay, the glucose and fructose, and their structure. Now we will look forward for their structures, okay. As we drawn their structure of glucose and fructose, when we took an example, okay, that fructose is a ketone group sugar and glucose is an aldehyde group sugar, okay. So now the important is glucose and fructose. So let's start to do the structure of glucose, okay. Let's try to do the structure of glucose. So uh, let's try to do about glucose. Okay, so glucose. Firstly, see, the glucose is also known as the blood sugar. Okay, blood sugar. It's known as blood sugar because it is transported to different parts of body through blood. Okay, so that's why it's known as blood sugar. Transported by blood. Okay, so that's why it's known as blood sugar. Okay, so and also it's known as grape sugar. It's also known as grape sugar. It's also known as grape sugar. So why I called it grape sugar is the reason is because all the fruits have fructose as a stored sugar. Okay, only the grapes have the glucose as stored sugar. Okay, only because only grapes have glucose as stored sugar, not fruit, not fructose, not fructose. Okay, but in other fruits, all other fruits, the fructose is the stored form of sugar. Okay, so let's move towards the structure of glucose. Structure of glucose. We can see their structure in two forms. Okay, we can see their structure in two forms. The first one is furanose, furanose, and pyranose. Pyranose structure. Okay, let's see what is furanose. Furanose is a five-membered ring. Okay, five-membered ring. So when glucose is present in the form of five-membered ring, then it is known as furanose, and when it is present in the form of six-membered ring, then it is known as pyranose. Okay, so I'll draw the structure of that ring. It's having one oxy group here, it's an oxygen. Okay, so one, two, three, then here four, and then five, and then six. So 4, 5, and 6. This is a 6 carbon. Okay, it is containing 6 carbons, and the membered chain 5 carbon, sorry, 5 membered chain, 1, 2, 3, 4, and 5. 5 membered chain is furanose, and if we talk about pyranose, then it will be something like this. So and they will be contain like connected multiple hydroxy groups, okay, and their position will be different, okay. Pyranose, okay, and this is furanose. So the second type of structures are their anomeric forms. Anomeric forms. Anomeric forms. So what do I really mean by anomeric forms? On the first carbon, it's the first carbon, second carbon, third, fourth, fifth, and sixth. So on the first carbon, on the first carbon of glucose, on the basis of the position of hydroxy group, either it is on the upper side or on the lower side, okay. So on the basis of them, the glucose is classified into two forms, which are called as anomers, okay, due to the position of these hydroxy groups. So I'll draw the structure again and we'll try to show you. It's first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. Yeah, okay. So whenever the hydroxy group of first carbon is present on the upper side, okay, when the hydroxy group of first number carbon is present on the upper side, and when it is present on the lower side, okay. So when it is present on the upper side, then it is known as beta beta glucose okay. And when it is present on the lower side, then it is known as glucose alpha glucose, and also alpha D glucose alpha D glucose. The D form of glucose, the D form of sugars are present in our body, not L form of sugars are not found in our body, and also not utilized in our body. Only the D form of glucose or other sugars are utilized in our body. So alpha D glucose, beta D glucose, we will like learn and will not be confused whenever we will see them as D or something written. And D means dextrorotatory. D means the dextrorotatory form, okay. Dextrorotatory form is the like it's something optical phenomenon, okay, when like some light is passed through the glucose, and it shows the rotation of that light. So it's some chemistry like concept, so we will not move towards it, okay. So it's D means dextrorotatory. So alpha D glucose and beta D glucose, whenever the hydroxy group of first carbon is present on the upper side, then it is known as beta. And whenever the the hydroxy group of first number carbon is present on the lower side, is known as alpha, okay. So these both are alpha and beta are anomers of each others, okay. These are anomers of each others, okay. So this was the structure. Now let's also try to compare the structure of glucose with fructose, galactose, and mannose also, okay. So let me draw their structure firstly. So uh, as I have drawn all the structures of glucose, galactose, mannose and fructose, okay, so we will try to see the difference between them. This is the glucose, this one is glucose, this one is galactose, and this one is mannose, and this one is fructose. Firstly, see the difference between firstly try to look at the structure. Okay, firstly we will see the position of hydroxy group. See, the hydroxy group on this one is first number carbon, this is second number carbon, this is third number carbon, this is fourth number carbon, and fifth and sixth. Okay, see, second number carbon, fourth number carbon, fifth number carbon, all are present on the right side. Only the third number carbon is present on the left side. Okay, so this is the structure of glucose. Okay, and on the basis of on the basis of fifth number hydroxy group, on the basis of position of fifth number hydroxy group, we name them as D glucose, because the hydroxy group is present on the right side. If this hydroxy group is present on the left side, then it is known as L glucose. Okay, levorotatory glucose. But that form of glucose is not found in our body. Okay, only D glucose is found in our body, and which is characterized on the basis of the fifth number of hydroxy group. Okay, on the basis of fifth number of hydroxy group. So in glucose, in the structure of glucose, or the carbon chain of glucose, on the third on the third number of carbon, the hydroxy group is present on the left side, and all the hydroxy other than third carbon are present on the right side. Okay, so this is the general structure of glucose. So we will compare the structure of glucose with galactose and mannose and fructose. Firstly we will see the difference between D-glucose and D-fructose. Also see, they are also D-galactose, D-mannose and D-fructose. Why? Because their fifth number carbon is present on the right side. Okay, so now the difference, everything is like similar in between their structures, only a single point is different. Okay, we will just find out the difference between them, and I have also highlighted the difference. See, in D-glucose and D-fructose, the difference is only of the group, the functional group. Here is the aldehyde group present, and here is the ketone group. The position of hydroxy groups are similar. See, on the third number carbon, hydrogen is on the right side and hydroxy group is on the left side. Then again fourth, fifth hydroxy groups are present on the right side. Okay, so so what's the difference? Only the difference is of the ketone group between glucose and fructose. Okay, so and if we see the glucose structure, difference between glucose and galactose, there is difference that uh, their third number carbon is having the similar position of hydroxy group. Okay, but there is difference in another number of carbon, or there is a difference in the, or change in the position of another hydroxy group of another carbon of these like carbohydrate molecules. Okay, see here in galactose, sorry, first, second, third, fourth and fifth and sixth. So there is difference in the position of fourth number of hydroxy group. Okay, the fourth number of hydroxy group is present on the left side. Okay, and in mannose, the hydroxy group of second number carbon is present on the left side. Every other than these specific hydroxy or positions, everything is just similar to glucose. Okay, so this is the difference between their structure, that the hydroxy group, that the hydroxy group of second number carbon is placed on the left side in case of mannose, and the hydroxy group on the fourth number carbon is present on the left side in the case of galactose. So this is the difference of glucose, mannose, fructose with glucose structure. Okay, so it was about glucose. So we have discussed the structures of glucose, mannose, fructose, and galactose now. And the very important question that arises here is the epimeric forms. Okay, which sugars contains like the examiner is gonna give you two names. Okay, the examiner is gonna give you two names, and he will ask you which of the following sugars are epimers of each other. So we know the epimers are glucose and mannose are epimers of each other, of each other epimers of each other, and glucose and galactose are epimers of each others. Okay, because the number of carbons and the number of oxygen and hydrogen are same in glucose and mannose and galactose. Just the difference is the position of hydroxy group. Okay, just the difference is the position of one hydroxy group. Okay, uh, 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 so the glucose and galactose are epimers of each other. The mannose and glucose are epimers of each other. Okay, but the mannose and galactose are not epimers of each other because there is difference of more than one hydroxy group. Okay, if we compare them, then there is difference between the fourth number and second number. Okay, the in the case of galactose, on the second number carbon the hydroxy group is on the right side, but in the case of mannose it's located on the left side. But and same as for the fourth number hydroxy group, which is present on the left side, but in the case of mannose it is present on the right side. Okay, so glucose and mannose, you will remember this thing very, it's very important. I give you as many as stars as it could be possible. Okay, so it's a very important point which can be asked. Okay, and also which one is a aldose sugar and which one is a ketose sugar. So fructose is a ketose sugar, and glucose, galactose, and mannose all are aldose sugars. Okay, so this kind of question is generally they are gonna ask you. Okay, so the next thing I think we should move towards oligosaccharides, and in oligosaccharides, second one is disaccharides. Disaccharides. Okay, disaccharides is a part of oligosaccharides. We can say is a part of, or is a type of oligosaccharides. Okay, for the disaccharides contains two units of monosaccharides. Okay, two monosaccharide units. Disaccharides contains two monosaccharide units. And if we talk about their general appearance, they are crystalline in appearance. Okay, they are crystalline in appearance, and water soluble. They are crystalline, water soluble, and sweet in taste. Okay, they are taste, sweet in taste. And if we talk about their examples, if we talk about their examples, the examples are, the first one is maltose. sugar. Okay, the second one is sucrose, and the third one is lactose. Okay. Disaccharides can be classified into two types. Okay, the it's very important, and you need to learn them by heart. There are two types. The first one is reducing disaccharides, reducing, which shows reducing nature, and forms osazone. Okay, osazone is some chemical compound which is formed by the reduction of these disaccharides. And the another one is non-reducing sugars, non-reducing, non-reducing one. Okay, the maltose and lactose are reducing sugars. Okay, and the sucrose is the only non-reducing sugars. Okay, the sucrose is the only non-reducing sugar. Okay, so you'll learn it by your heart, you'll have to remember it anyhow. So you'll have to remember it. So reducing sugars are the maltose and lactose. These are reducing sugars, and sucrose is the only non-reducing sugar. Okay, sucrose is the only non-reducing sugar. And there what is that thing that explains the reducing nature of any carbohydrate? So the free reducing nature of any carbohydrate, just a second. Let's write here the reducing nature of any carbohydrate, any carbohydrate is due to its free aldehyde or ketone group, free aldehyde or ketone group. So carbohydrates shows their reducing behavior due to their free carbohydrate, free aldehyde or free ketonic group. Okay, so free, yes. Okay, so I'm gonna make it in the box. So this was about disaccharides generally. Now we will talk about maltose, lactose, and sucrose separately. Okay. So let's start to learn about maltose. Maltose is formed by the combination of two glucose. Okay. Maltose is formed by the combination of two glucose units. I have already drawn to save the time. Okay. So look here. This one is a glucose structure. Okay, alpha D glucose. Alpha, why did I write alpha? As you can see, the hydroxy group is on the lower side, so it's named as alpha. And if the hydroxy group is on the upper side, then it is named as beta. Okay, and D, why did I write D? As we discussed before, the D is written due to the fifth number hydroxy group, which is in this structure you will not see, because the fifth number hydroxy group is now like included in the formation of this ring chain. But when it is in a straight carbon chain, then it is written as it's found on the right side, and the first carbon and water is eliminated and the formation of alcohol, somewhat this process happens. Okay, so we don't discuss here this thing. So we know that D is written due to the location of fifth number hydroxy group of glucose on the right side. That's why it's alpha D glucose. You can say alpha glucose or alpha D glucose. It's better to say alpha D glucose, but if you like don't remember to say D, so it's okay. And the examiner can give you in either forms. Either he can give you alpha D glucose, or alpha D, or alpha glucose, or alpha glucose. Okay, so you can say anything. So let's write the forming units of maltose is alpha glucose plus alpha glucose. Okay, the forming unit, you have to remember this point. Only these are the important points here: the forming units and the linkage. Okay, the examiner is gonna ask you the forming units of maltose and lactose and fructose, and linkage between them, the bond formed between them. Okay, so let me clear you this concept also, the how the bond is formed. Okay, so just a second please. uh, it's an OH. So see, this one is the first number carbon, the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and this one is the sixth carbon. Okay, so the first number carbon, first hydroxy group. Okay, the hydroxy group on first number carbon of one glucose, and the hydroxy group of another glucose which is located on the fourth number carbon. Okay, from first and fourth the water is eliminated, water is eliminated. Okay, the water is eliminated, and it leads to the formation of one glycosidic bond, one glycosidic bond. Okay, this one is the glycosidic bond, and as it is formed between the alpha 1, okay, the first number carbon and fourth number carbon, so it is named as alpha 1 4, because the first number carbon is involved and the fourth number carbon of another glucose is involved. So this is known as alpha 1 4 glycosidic bond, glycosidic bond or linkage. Okay, so the linkage which is which linkage is formed here, alpha 1 4, because both of the sugars were alpha. So the alpha 1 first number carbon of one glucose and the fourth number carbon of another glucose, so the linkage here formed is alpha 1 4 glycosidic linkage. Okay, glycosidic linkage. So you'll have to remember this thing. Okay, you'll have to not only remember, you'll have to learn by your heart that maltose is formed by two alpha glucose units, and the bond between these two glucose units is the alpha 1 4 glycosidic bond. Okay, only this question is gonna they are gonna ask you. Okay, so it was the okay, and they are further hydrolyzed by, further hydrolyzed by an enzyme, maltase. Okay, the enzyme will act on this structure, the maltose, and will add again H2O will be added here, and it will again leads to the formation of two glucose units. Okay, so it is hydrolyzed by maltase. Okay, and also we know that it is a reducing sugar. Okay, it shows reducing property. Why do it shows reducing property? Because it is having a reducing carbon. Okay, it's having a reducing, and we can say it's one. So in the previous slide, we saw the reducing nature of a carbohydrate is explained by the free aldehyde or free ketone group. Okay, is explained by the free aldehyde or free ketone group. The carbohydrate which is having one free aldehyde, either it's having free aldehyde if it's in the it is in the form of this like not in the form of ring. Okay, it is it's if it's not in the form of ring chain, okay, then it's free aldehyde group shows the reducing property. But if it is in the ring chain form, okay, if it is in the ring chain form, then the carbon of that aldehyde group, the carbon of that aldehyde group should be free, and then it shows the reducing property. So we can see here the like one glucose is not having its carbon free, one glucose is not having its first number carbon, which was connected like which was annotated as aldehyde group. So it is not free, it's bonded with the fourth number carbon of another. But the another glucose is having its first carbon free, so the first carbon will show the reducing property of that sugar. Okay, so I have also noted it here especially to remember, to make you remember always. So the first carbon of glucose or aldehydic carbon, okay, the first carbon of glucose or aldehydic carbon, okay, the carbon which was in the form of aldehyde. Okay, so in the case of glucose, the first carbon of it, or the aldehydic carbon, or in the case of fructose, in the case of fructose, when we will see the non-reducing nature of sucrose, we will see the structure of fructose. So in that case, the carbon which is the ketonic carbon, okay, is not free. Okay, so in case of fructose, where the second carbon or the ketonic carbon must not be a part of glycosidic bond, then it shows the reducing nature. But here it's only because as one carbon is free here, okay, so it shows the reducing nature. So just remember this point, and I'm also like putting this uh, note in the box, okay, just to make you remember, and just to highlight it. So yes, so just remember this thing, why these sugar shows reducing property or non-reducing property, because uh, either the first carbon of glucose, galactose, or glucose, of glucose or galactose, or some others. Okay, should be free. And in case of fructose, the second number carbon, as we talked in previous slides. Okay, the second number carbon which contains the ketone, which is annotated as ketone group. Okay, should be free. Okay, either the ketone group should be free, either the ketonic carbon should be free, so we can say anything. So just now it's sucrose. Sucrose is also known as cane sugar. Sucrose is also known as cane sugar, because sucrose is the stored form of sugar which is present, which is stored in the sugar cane. So that's why it's known as sugar, cane sugar. Okay, so it's known as cane sugar, and as we talked before that it shows non-reducing property. The sucrose shows non-reducing property, non-reducing. We can say non-reducing sugar. The sucrose is the only non-reducing sugar. You will always remember this thing. Non-reducing sugar, non-reducing sugar, and as it doesn't show reducing property, so it will not leads to the formation of osazone, which I talked before. It's some chemical compound when some carbohydrate shows reducing property. Okay, so and can't form, and so can't form osazones. Uh, if we talk about the forming units, so the forming units are alpha D glucose, alpha D glucose, as you can see here, and beta D fructose. Okay, beta D fructose. So see here, alpha D glucose and beta D fructose. Why I said beta? Because fructose, the first number, you will see the first number. You can say alcohol group is on the upper side, and this one is on the lower side. Okay, so this is beta D. I explained you before why I am saying D, beta D fructose, beta D fructose, and glucose is alpha D glucose, alpha D glucose, because the hydroxy group is on the lower side. Okay, so beta D glucose, beta D fructose and alpha D glucose, and the linkage here formed is C. The first number carbon of the glucose and the second number carbon of the fructose is involved. So it's alpha 1 combines with the beta 2, alpha 1 beta 2 glycosidic bond, alpha 1 beta 2 glycosidic linkage. Understood? Because the first number carbon of alpha glucose and the second number carbon of beta fructose is involved in the formation of that glycosidic linkage. So that's why glycosidic bond. Okay. So that's why it's known as alpha 1 beta 2 glycosidic linkage. And the important points which are here are which one is known as the cane sugar, sucrose is known as the cane sugar, which is the non-reducing sugar. Sucrose is the only non-reducing sugar, so I named it as 5 star. Okay, I have given it 5 star. And the forming units also you should remember by your heart. 5 points. Okay, 5 star. And it is also a sweetening agent in food industries. It is used as sweetening agent. Okay, so you can say artificial sweetener is the sucrose. Sucrose is the artificial sweetening agent. Sucrose is the artificial artificial sweetening agent. Okay, used in industry. And the hydrolysis, sucrose is again hydrolyzed by the enzyme sucrase. Sucrase, or it is also known as invertase. Invertase enzyme. Okay, either by sucrase or either by invertase. Invertase, because sucrose is also known as invert sugar. And why is it known as also known as invert sugar? Because when it is hydrolyzed, then there is a mixture of glucose and fructose. So the mixture of alpha D glucose and beta D fructose, okay, is known as invert sugar, is known as invert sugar. So that's why it's also known as invert sugar, because when it gets hydrolyzed, so we get the mixture of alpha D glucose and beta D fructose. So that mixture is known as actually invert sugar. Okay, so this was about sucrose. Now let's move towards lactose. Lactose is also commonly known as milk sugar, and because it's the common sugar which is found in the milk. Okay, the forming units of lactose are beta beta D galactose, galactose, and beta D glucose, beta D glucose. And the linkage present between them is beta 1 for glycosidic linkage. Okay, the linkage between them is beta 1 for glycosidic glycosidic linkage, acidic linkage. Okay, and it shows the reducing property. Why do it shows reducing property? Because of the free first number carbon of glucose. Okay, because of the first number carbon of glucose is free, or you can say the aldehyde carbon is 3. Okay, you can see here. Understood? Where I am pointing. So uh, it exhibits reducing property. Okay, so from these, I think we clear every important point, whatever could be possible. Okay, so just you have to focus on these start points. Okay, this will be commonly asked from the structure. So now we completed oligosaccharides, the important part of oligosaccharides. Okay, the other other parts of oligosaccharides, which were trisaccharides, intermediates, and tetrasaccharides, were not so important for us. So now polysaccharides. Polysaccharides are macromolecules. Macromolecules means molecules which are having large size. Okay, so polysaccharides are macromolecules which are composed of more than 10 monosaccharide units. Okay, which are composed of more than 10 monosaccharide units, and they are further classified into two main categories. One is Homo Polysaccharides, and then another is heteropolysaccharides. Okay, so the first category of these is Homo Polysaccharides. The first one is Homo Polysaccharides. Homo Polysaccharides, and then second one is heteropolysaccharides. Heteropolysaccharides. Okay, so the examples of Homo Polysaccharides, or the like carbohydrates which are included in uh, Homo Polysaccharides, are cellulose. The second one is starch, glycogen, chitin, and inulin. The most important one. Okay, I just wrote the most important one. Okay, so cellulose, starch, glycogen, chitin, and inulin. And in the case of heteropolysaccharides, the carbohydrates included are uh, first one is high hyaluronic acid. Okay, the first one is high hyaluronic acid. The second one is Roitin, chondroitin. Okay, chondroitin. And the third one comes heparin. Fourth, fourth one is Dermatend Sulfate. And the fifth and last one is Chitin sulfate. Chitin sulfate. Okay. So let's move towards the cellulose. Okay, firstly we will see the first part of this, which is Homo polysaccharides. And from Homo polysaccharides, we are looking for firstly cellulose. Okay, the cellulose is a long chain. Okay, and it's unbranched, which are composed of beta glucose subunits. You can see two glucose subunits which are having the first hydroxy group on the upper side. Okay, and the bonding between them, or the linkage between them, is found between the first carbon of one glucose and the fourth number carbon of the another glucose. Okay, so many of these glucose subunits or units are attached together by beta 1 for glycosidic linkage. Okay, and this cellulose acts as the main structural polysaccharide in plants. Okay, and the fibers which we eat, okay, the plants or the products of plants which we eat, their fibers plays a very important role to make our digestive system very healthy, and also they helps in the bowel movement. Okay, so this is the main important role of cellulose in our body, or and you'll have to just remember the bonding on the subunits. Okay, so the another one is the starch. Okay, the starch is the polymer of alpha glucose subunits, and it acts as the storage polysaccharide in plants. It acts as a storage polysaccharide in plants, the storage form of carbohydrate, which like the carbohydrate which is the stored in plants. Okay, so it is the stored form of the carbohydrates in plants. Okay, so starch can be found in two forms, either in amylose or either in amylopectin. Okay, so amylose and amylopectin, the difference you can, as you can see, is of branching. The amylose starch, the amylose form of starch is unbranched. Okay, it's a linear chain or helical. It's in helical form. Okay, so and whereas the amylopectin is also helical but is branched. Okay, and as you can see, uh, the one end of amylose is reducing one, the which one I marked like tick mark, and the another which I marked with cross is non-reducing end. Why is it so? So I have drawn a structure of amylose in which it's having alpha glucose subunit, one alpha glucose subunit, another, and so on. Okay, so as you will see here, the first number carbon of one alpha glucose subunit and fourth number carbon of another alpha glucose subunit, which are both alpha, means they are both having their OH group on the downside. Okay, on the lower side. So water molecule is removed from here. Okay, and then alpha 1 for glycosidic bond is formed. Okay, so you will see, as this like chain continues to be formed, the first carbon of alpha, this glucose is free. Okay, wherever it will end. Okay, the first carbon is always free, or the aldehydic carbon, we called it before. Okay, so it's always free, so that's why it's the reducing end, and I have marked it as the reducing end. Okay, so let me also write here that the tick uh, uh, this one is the reducing end. Okay, and the another one is the non-reducing end. Okay, in the case of amylopectin, you will see that this is the non-reducing end and this is the reducing end. Okay, and this is the non-reducing end and this is the reducing end. Like the branching which is formed in the case of amylopectin is formed towards non-reducing end. Okay, means firstly for branching, first number carbon is attached. Okay, which one is attached? First number carbon. First number carbon of the glucose, alpha glucose, is attached to the sixth number carbon of another glucose for branching. So that's why this is it was the reducing end. Okay, and this is non-reducing. So you can say that in case of amylopectin, there in branching there is no any kind of reducing end. Okay, so I have drawn the structure This one is the alpha glucose, one alpha glucose and another alpha glucose. So in parent chain, not in branching, in parent chain, you will see the bonding is occurring between the first carbon of one glucose and the fourth number carbon of another glucose. Okay, this is the carbon of fourth number carbon of another glucose. So the linkage is alpha 1 for glycosidic bond, and you will have to remember it, which type of bond linkage is present in parent chain, okay, in parent chain, and which type of linkage is present in the branch of amylopectin or whatever. Okay, so alpha 1,6 glycosidic bond is formed in branching of starch, okay, in branches of starch, and they don't show any kind of reducing properties. Branches of starch don't show any kind of reducing properties. Okay, only the parent chain shows the reducing properties. So now let's move towards another polysaccharide, which one is glycogen. Okay, the glycogen is the storage form of sugar in animals and humans. Okay, the stored carbohydrate in animals and humans are very important. Okay, in our body, the carbohydrates are stored in the form of glycogen. Okay, so and if we talk about their structure is obviously similar to amylopectin, which was having branches. Just the difference between their structure is that glycogen is having more and more number of branches rather than amylopectin. Okay, so it was there was 2 points which were very important on the basis of exams. Okay, so now it comes to the chitin. Okay, chitin is a polymer of nag means n-acetyl glucosamine. Okay, is a polymer of it's a sugar. Okay, so it is present in fungal cell wall. Okay, the chitin is the main component of cell wall of fungi. Okay, in microbiology we study this structure. Okay, the nag that is present in the cell wall of also in some bacteria also it is present, and in chitin, sorry, fungi also. Okay, so present in fungal cell wall, and also it is present in exoskeleton of arthropods also. Okay, so these two are the main important points, and also for people or for students who are preparing for NEET. Okay, this point is very important for them. Okay, so the next point or the next homo polysaccharide comes is inulin, which is very important. Okay, so it is a polymer of beta D fructose, and the bond formed here, you just need to remember 3 star to it and 2 star to it. Okay, and it is the indigestible homopolysaccharide, and it is also water soluble. So that's why it is injected in the blood system. Okay, it is injected in the blood system, or maybe can be given orally for measuring the GFR, glomerular filtration rate. Okay, and you will study the whole mechanism, or like about it, everything about it in the physiology. Okay, that inulin is given orally or by intravenously, because it is water soluble and it is non digestible. Okay, so that's why it is completely removed by the glomerular filtration. Okay, so by some mechanism or by some physiological phenomenon, we can conclude that how much is our glomerular filtration rate is going on. Okay, so inulin is very important in physiological part. Okay, so now it comes to the heteropolysaccharides. In heteropolysaccharides, we are looking for firstly hyaluronic acid, which is a polymer of D-glucuronic acid plus NAG. Okay, two main subunits are forming this polysaccharide. One is D-glucuronic acid and the another is NAG, N-acetyl glucosamine. Okay, so The importance of hyaluronic acid is that is it is present in joints and acts as lubricating agent, and as well as it also acts as a shock absorber. It also acts as a shock absorber in joints, as well as a shock absorber 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 okay, in joints. Okay, so and it is also it also acts as animal cement. Why do I call it animal cement? Because it helps to connect cell walls. Oh sorry, because it helps to different connect different cells. Okay, it helps to connect different cells. So and it is also present in vitreous chamber of our eye. Okay, and it also covers the ovum. Okay, and an enzyme hyaluronidase, enzyme hyaluronidase, uh, just a second, enzyme hyaluronidase, hyaluronidase. Okay, which is concentrated in seminal vesicles. Okay, no, sorry, which is concentrated in testes and then is collected or in the seminal vesicles and secreted through the seminal fluid. Okay, it is very helpful in the fertilization, because during fertilization hyaluronidase acts on the hyaluronic acid layer of ovum and then degrades it, and then helps to the sperm to move further inside the ovum and then fertilize it. Okay, so an enzyme that is hyaluronidase. Okay, so hyaluronidase. Now the next one comes here is the chondroitin is also known as chondroitic sulfate or chondroitin sulfate. Chondroitin sulfate is the ester form of chondroitin. Okay, so it is present in cartilages. Okay, tendons and bones also, but in cartilages it is present in much more amount, and tendons it's just normally, and in bones it's in really in very less amount. Okay, so the polymer of it is the polymer of D-glucuronic acid and NAG. So these were important points for chondroitin. Okay, so now next comes heparin is a very important compound used in medical field, which acts as anti blood coagulant. Okay, and the next one comes is the next one which comes is Dermatend sulfate. Okay, dermatend sulfate, the structure of dermatend sulfate is just similar to the chondroitin sulfate. Okay, it is composed of ioduronic acid and NAG subunits. Okay, it maintains the shape of tissues. It helps in the maintaining of shape of tissues. Okay, the in our like skin also. Okay, sulfate, if we talk about keratin sulfate is composed of D-galactose and NAG subunit. Okay, and it keeps our cornea transparent. The transparent structure of our cornea is due to keratin sulfate, a large amount of keratin sulfate. So guys, this was all about carbohydrates, general structure, their classification, their examples, and their bondings. Whatever the important part which we should focus in carbohydrates and their structure is just the examples, the names of carbohydrates, and what in which like category are they included. Okay, are they monosaccharide, disaccharide, or polysaccharide? You'll just have to learn them by heart, and also the bonding between them. In the case of disaccharide or polysaccharide, you will have to learn the bond, which bond is formed like which bond is formed during the branching of any polysaccharide. Okay, or which bond is formed between two like in between the subunits of sugar units. Okay, so just these things are generally asked. Okay, and also I will like try my at my level best to arrange whatever we did here, okay, to provide you PDFs so that your time could be saved during you should not write these notes. I will provide the link of this PDF, okay, you can just download it from there, okay. So guys, if you like this chapter, okay, and you understood very well what I tried to make you understood, okay, so please like, share and subscribe to my channel if you like do it yet. So please subscribe it, and uh, uh, we will meet you in the next video with carbohydrate metabolism. Till then, thank you so much, take care, bye bye.